Welcome back. This is a continuation of 18.1. So this is part 2. I'm going to pick up where I left off with the first video, and that's talking about the trip operon and how it operates. To start with, operons are controlled by a promoter, which allows RNA polymerase to bind, which will then transcribe the genes of the trip operon, which will produce enzymes, which are the E, D, C, B, and A. These enzymes will be used in a metabolic pathway to build the amino acid tryptophan. The promoter allows RNA polymerase to bind to it only if the operator is active. And by active, what I mean is there is not a repressor bound in place to it. If there's a repressor in place, RNA polymerase cannot bind to the promoter, and transcription will not happen. Now, what controls the regulation of the repressor being inactive or active is a secondary gene called the regulatory gene. A regulatory gene will produce some mRNA that controls the functionality of the repressor. If the regulatory gene is not active, the repressor will be inactive and it cannot bind to the operator. In this case, the polymerase will transcribe the genes of the trip operon and ultimately will have tryptophan produced. However, if the regulatory gene is active, it will produce mRNA, which will bind to the protein, the repressor, causing the repressor to become active. When the repressor is active, it has a co-repressor, which is the presence of tryptophan, and the combination of mRNA and tryptophan, the co-repressor, will bind in place, activating the repressor, and it will bind to the promoter, which will, as a result, prevent RNA polymerase from binding, and no more tryptophan will be made. So in summary, the repressor is active when tryptophan is present and in good quantity. The regulatory gene is always producing the mRNA right here, so this is always present. It's just a matter of, is tryptophan present? If tryptophan is present, this becomes active and no more tryptophan will be produced. So moving into question 10, which is talking about repressible and inducible operons. There's two types of operons. There are the repressible ones, which are usually on but can be turned off, and there are inducible operons, which are usually off but can be turned on. So they're the exact opposite of one another, functioning until it's turned off, not functioning until it's turned on. The trip operon happens to be one that is repressible, and we saw that in the previous slide, where it's on, and then when it produces enough tryptophan, that co-repressor, it will bind to the repressor, and then it will turn off the operon. So to complete our response to number 10, it's asking for some examples of each. The trip operon is a repressible one, and the LAC operon, L-A-C, LAC, is an inducible one, which we'll be talking about quite shortly. That's actually number 11 on your reading outline. Here we'll talk a little bit more about that LAC operon, L-A-C, LAC operon. Once again, it is inducible, meaning that it is off, but it can be induced, it can be turned on. And that's done by what is called an inducer. And an inducer inactivates the repressor to turn the operon on. So we're looking at number 11 on your paper, which this image kind of compresses or expands everything that's in your image. Your image is looking specifically at this bottom part. And since it's a little choppy, I'll help you fill in through the numbers here. So on your paper, number 1 is pointing to this region right here, which is the regulatory gene of the LAC operon, LAC1. So a regulatory gene is number 1. Number 2 is pointing to this red thing on my sheet here, which we are going to title as the repressor protein. Repressor protein is number 2. Number 3 is pointing to the circle right here, which is going to be our inducer. Number 4 is pointing to this bubble, which is really hard to see on your paper, but number 4 is RNA polymerase. Number 5 is pointing to the strand, which is going to be mRNA. Number 6 is pointing to this entire region right here, which is going to be the lac operon. You might also want to tie in the operon genes underneath it, which the operon genes are actually number 7. So the entire thing, number 6, is a lac operon, and then number 7 is pointing specifically to these, which are the lac operon genes. Number 8, skip. Don't do number 8. But what I would like you to do is there is an arrow that is not numbered, and that is pointing to this item right here, which, if you recall, that is our operator. So please label the operator as well. So the lac operon is an inducible operon, meaning that it's usually turned off. So here's how this one works. We're going to look at the top part of the image from the previous slide, and then we'll move to the bottom image. This is showing us what is happening when lactose, which is why it's called the lac operon, it makes lactose, a sugar. When no lactose is present, the suppressor, or repressor I guess, is active, meaning that it will bond in place, and we cannot bond the RNA polymerase to make it. So the repressor is active and our operon is off. However, when lactose is present, lactose, the inducer, will bond to the repressor protein, inactivating the repressor protein, and it will then jump off of the promoter, the operator here, and RNA polymerase will then be able to bind to it. So the presence of lactose will bond to the protein, causing it to become inactive, and now our operon is turned on. Number 12 is asking you to compare and contrast the lac operon with the trip operon. I'm going to let you try that one on your own. You might want to go look at the book for some assistance with this, or you can review what we had discussed previously. But think about how they're similar and think about how they're different. Questions 14 through 16, which we'll be covering on the next 3 slides here, so it's not all right here, deals with 2 items: CAP, which is short for catabolite activator protein. It is an activator of transcription. And CCAP, or cyclic AMP. So question 14 is asking what CAP is and how it works. Your answer is right here for the first part. It's an activator protein that positively controls the operon. How does it work? It works when glucose is scarce, which will cause CAP to be activated by bonding with cyclic AMP. So when activated, CAP attaches to the promoter of the lac operon and increases the affinity or ability to bond to RNA polymerase, and this accelerates the process of transcription in that lac operon. So how CAP works is when glucose levels increase, CAP will detach from the lac operon, and that will return transcription to a normal rate. So CAP is said to be a form of positive feedback, meaning that it will increase the rate that something is occurring, in this case transcription. Sorry about the bell. We can see that CAMP, or cyclic AMP, will bond to our CAP, and it's inactive right now, but with CCAMP or CAMP bonded in place, now it becomes active. It will bond to the promoter, and in this case it will allow RNA polymerase to bond, which will then cause transcription of the lac operon to occur. So this is when lactose is present and glucose is scarce. But on that previous slide, what happens when glucose is present? Well, now CAP becomes inactive, and we cannot have any transcription occur. Number 17 is another one that I'd like you to look at on your own. The question is, how can both repressible and inducible operons be negative regulators? So what I want you to think about is the trip and the lac operon. How is it possible that they are both negative regulators when one is repressible and the other is inducible. Think about that. How is it possible that they are both negative regulators when one is repressible and the other is inducible? So number 17, I'm putting on your shoulders.